After moving on to that, external force is realized into the mechanical forces like ultrasound, microwave, high pressures, pulse electric fields, pulse light, everything at the innovative extraction process, which is known as the innovating or emerging technology for the extraction. Today's is basically for the extraction of bioactive compound using ultrasound as well as the microwave. Okay, have you have already known as the bell leaf you are already cordial with. This is a very nutraceuticals and pharmaceutical, also a cosmetic importance in the bell leaves. This is the bell leaf powder. Okay, the sample is we will use is the bell leaf powder. It has been dried, ground and sieved properly in a constant particle size, so that our extraction procedure will be uniform. Okay, then comes to the first, we'll explain about the ultrasound assisted extraction. The ultrasound assisted extraction is involves the ultrasound wave. What is the ultrasound waves? In the sound wave you are particular about, it is the part of a sound wave which is not in the we cannot audible. It is because it is beyond our human hearing. Human hearing is between the 20 hertz to 20 kilohertz, but the our ultrasound wave is above the 20 kilohertz, so it cannot be audible to you. Then it is the mechanism of the extraction process is that a means a term that is the acoustic cavitation. What is the meaning of that? Acoustic cavitation is the formations, growth and collapse of the microbubbles forms inside the solvent solution adjacent to the cell walls. Okay, what is the concept? Now, due to the electromagnetic field, electric fields, there has been the conversion of electric fields to the mechanical vibration, that is ultrasound waves. Ultrasound waves has oscillating fields, oscillating vibration. These oscillating vibrations or high pressure, low pressure conditions will makes the bubbles adjacent to the cell walls, micro bubbles. These bubbles will grow and after some times it will be collapse after some time, and after the collapse there is a high temperature will arise adjacent to the cell wall. You just imagine a cell wall, one cell wall. Okay, cell at, means adjacent to the cell wall, this is maybe the cell. Okay, this is the cell wall. Adjacent to the cell wall the bubbles are created due to the ultrasonic waves. Due to the heavy ultrasonic waves or high pressure, these bubbles will be collapsed, and after this collapse, thus the high temperature will rise. The temperature will disrupt the cell walls. After the cell wall will be ruptured, the solvent outside the surrounding the uh, cells, it will be means put inside the cells, and it will mix with the bioactive compounds or target compounds inside it. And then this will be dissolved into the solvent surroundings. Got it? Then in the actually these ultrasound waves, we are putting the external force as a ultrasound waves. So, because in conventional extraction is mainly about the mechanical stirring. Overnight or 24 hour or 48 hours you have to stir it to solubilize the bioactive compound present inside the cells to outside the solvents, like this only. But but what is the main mechanism inside the ultrasound? It will eject the, but it will dissolve the bioactive compound in a very less time with a extra force. That's why it is taking less times, and it will be beneficial as compared to the conventional extraction. Got my points? Okay. Okay. Ultrasound extraction has two types. One is bath type and another is probe type. Okay. Bath. This is the probe type. Basically, I am telling you. This is the generator. Okay. This is the generator. This is the transducer. This is the probe या सोनोट्रोप भी बोल सकते हैं हम. Then this is the vessel stands, extraction vessel stand, and this is the our cooling systems. Cooling system in some extractor machines is be attached, but here that is not possible. So we are in manually we have to cooling by the ice baths. Got my point? Okay. First is the generator. Generator is that which converts the electric field which is given by the from the electric source or whatever electric field to the high intensity electric signals. It will also affects the intensity, power, amplitude and frequency of the ultrasound. At which power, at which frequency, at which amplitude you intend to extract, it will decide by the generator. You command to the generator how much amplitude do you want to give the high electric field signal, then it will be then it will give this types of means this amount of the electric signal to us. Then this is the transducer. Transducer is for the high intensity electric जो भी signals है, then it will convert these signals into the ultrasonic waves, mechanical vibrations को convert करता है. Then it will pass through the probes and which is directly contact with the vessels. And then when we'll commands to the generator that this amplitude at this time we will give to the extraction for the extraction, then it will convert the ultrasonic wave and it will pass through the probes, and from the probes it will give to the extraction vessels. At this time the extraction will be happen. Okay. How the cooling system is meant for the, because in ultrasound there are bioactive compounds. If you are willing to do the extract the bioactive compounds also be in volatile natures, and volatile natures cannot sustain up to means high temperatures. They need in the normal room temperatures. Switch on the light, switch on the ultrasounds. Okay. Cool down the whole extraction process so that our means the volatile compounds can be secured well. Okay, got me? Then we will prepare the sample. How? Means extraction. One is the preparation of sample, then is the extraction process, and second one is the separation or the filtration process. Just do the वो 2 ग्राम of जो बेल लीफ है उसमें 30 ml वाटर डालें. This is the amplitude based generator. Sometimes you can see also power based or the frequency based generator. Here the frequency and the power has been constant, and the amplitude you can vary. Okay. In amplitude, if it is taken as a 60%, the amplitude unit is the 60%, means you can also convert the amplitude to the power. Okay, that is the 60. It is the time, and it is the pulse on time and pulse off time. Pulse on means how much time you want to give the ultrasound, and pulse off is how much time you give it to the rest positions. Okay, this is the 60% amplitude. Process time is 20 minutes. Pulse on means 5 seconds you have to give ultrasound, and 5 seconds it is to be filled. Okay, so I am just I just give you some test about the 5 minutes. Okay, pulse on, so I will take as a 10 seconds. Okay, pulse up. Then we will put the extractor vessel inside the clump. Now I took the solvent as a water, because we all know that water is the universal solvent. Okay. Then you can take it as any polar, non-polar, any deputatic solvent, any type of solvent you can use for the ultrasound. That's why it is known as the very emerging technology. It can be versatility, any kind of polars, any kind of solvents you can use. I took 2 grams of bay leaf powder, dry powder, and 30 ml of water, because I have optimized already in the design software. It is a software, means in software you can how many independent variables you can took, and how many dependent variables you can take. On that basis you can optimize the condition. That condition is 60 amplitude at 5 minutes, and if 2 gram of sample you take and 30 ml water, you can take higher yield of the bioactive compounds. That has to be optimized before doing the ultrasound experiments. Okay. Okay, and I put the probe inside it. It should not touch to the bottom of the vessel so that it will be broken. Okay, then we will keep it as a close. Enter. It will start. Okay, so during the start I am telling you what are the applications, advantages and disadvantages. Okay, the applications would be consist of it can be used at after the extraction. You can purify and it can be used as any supplements and any type of food supplements, any type of functional food you can use. Then the health, I have told that health in pharmaceutical use, cosmetic use also, the so high anti-aging pigments, anti pigmentation, all these things you have heard of, that is also can be by the extraction of means bioactive compound can be done. Then environmental, how it can be affected, because in agricultural waste there are many types of means essential compounds will there, like in you have seen that sugarcane juice center, there are straws are the exposure or the disposal of the this juice center. You can see in that there is a very good amount of fibers in that. You can extract the fiber from the sugarcane. For example, coconut tender coconut soap, you can also extract fibers. Okay, then you can also find some the fruit cells, like pineapple and jackfruit. These cells have been be wastage, but it can be also used as the extraction of the starch. It has a very good amount of starch. Okay, and then after that if you intend to extract the protein, प्रोटीन के लिए भी बहुत सारे कंपाउंड्स, लीव्स, सीड्स and some means like fruits ka peels, all these are used as the proteins. And if you are intend to the gums, all these things, seaweeds is a very much now trending waste is there, which can be applicable for the extraction of gums, which can be used as a packaging of films, all these things. Got it? The advantages of ultrasound is that it is very efficient as compared to the conventional extraction, because in lesser time, in 5 minutes you can get good amount of yield, but whereas at 24 hour, 48 hour, the conventional extract is not given that much of bioactive compound. Okay, then higher yield is there, lesser time, very minimum time you can get your results. Okay, because time is the money. Okay, if you and the thing is environmental friendly is there, any type of solvent you can use. Okay, and the thing is the versatility I have already told that any type of solvent you can use in the ultrasound. Okay, there is no effect means affecting by the equipments. Then what is the disadvantages, any guesses, what is the disadvantages in the field? What's your ah that should be the there. Operators should be skilled enough to operate, means to demonstrate or to experiment these things. Regardless, ultrasound is very costly actually. Okay, in industrial scales it cannot be operated till time. Okay, because of the investmental cost is there, means capital cost is there, and also energy will be there, because it will take a high electric field, means where you have continuously electrical field, then it can be used and be there. Okay, the second one, what is that, second क्या हो सकती है, any guesses, in ultrasound what is the second parameters? Yeah, yeah, means while essential oils I am talking about, essential oil is very much volatile in nature. This much vibration or this much mechanical disruption also degrade these samples. For this, because टू means क्या बोलते हैं कि conventional extraction जो है उसको रोकने के लिए हमने ये इमर्जिंग टेक्नोलॉजी वी मेड, इमर्जिंग टेक्नोलॉजी लाइक दिस ओनली टू and to means to diminish its disadvantages, and some other novel extraction techniques also have been evolved, like cold plasma assisted extractions there, and pulse light extraction is there, supercritical carbon oxides extraction is there, subcritical fluid extraction. These are novel extraction types to diminish these disadvantages. Okay, this is not the optimistic extraction technology. It has some disadvantages. To overcome this, all these extraction techniques have been taken care. Got it? Okay. You can see that means before you have seen that the powder has not been dissolved fully, but here you can say that here it is clearly looking like a some means bioactive compounds or anything is in it. It has been dissolved into the solvent. After that it can be go okay. After this we'll centrifuge, and after that we'll find the clear solutions or solvents, and the will extract it above, separate above. Okay. Okay, we'll now next we'll go for the microwave assisted extraction. The microwave assisted extraction. Okay, microwave assisted extraction also be is a external force is a microwave. Microwave is the electromagnetic field. It is given as the means microwave is the electromagnetic field which is given to the food samples to extract the similarly bioactive compounds. But here the mechanism is different. Actually, when we will give the microwave to the means microwave power to the samples, then the alternative oscillating electromagnetic field is responsible for the extraction. How? Now here one disadvantage is there, only the polar compounds, only the polar solvent it can be used in the microwave, because in the oscillating or the in the oscillating electrical and magnetic field, the polar compounds या polar factors inside the solvent as well the samples can be interacted with the microwaves, only the polars and ions present in it. So non-polar compounds non-polar solvent cannot be used as in the microwave assisted extraction. That is the bigger challenge in the microwave assisted. Then what, there are two principles working on the for the microwave assisted extraction. One is the dipole rotation and another is the ionic conductions. Dipole rotation, what is meant by, when we will give the microwave to the food samples, due to the oscillating electrical fields, electromagnetic fields, the polar compounds, polar means hydrogen, oxygen, all these compounds will be aligned proportional to the oscillating fields. Got it? Means suppose this is the sine curve. Like sine curve, electromagnetic field is rotate means is given by. When it is in the upward field, this is the means the polar compounds align like this. Okay. Then when it is the downward field, the polar is this. Okay. This is continuously, when we continuously give the microwave, then the polar compounds like hydrogen, oxygen, they will be charging like this, they will be oscillating very fast, and it will create the heat. Okay, it will very means high temperature it will create. Okay, these high temperatures is responsible for the disruption of the cell walls. Imagine this is the cell wall, it breaks, the high temperature breaks the cell walls, and the solvent the target compound present in it, it is dissolved by the solvent. Okay, then it will be by the penetration of the solvent, then it will means all the solubilization happen, and all the target compound is been released. Got it? Then we'll go and see the how the extraction will be happen. And basically in the microwave, here you can take glass or any type of material, the extraction vessel can be, but in microwave there should be a specific vessel for the extractions, because there are specific vessels to need to be there, because the microwaves should be penetrated throughout the samples. Okay, so glass is the major compounds or major vessels, so that you can means the microwave can penetrate it through this. Okay, like this we have to pour all these things same. Okay, I'm just giving 30 ml of water. Okay, yeah, same thing. Just go to microwave. हाँ, उधर, हाँ उधर जाके अंकन को बोलो कि वो, okay, we'll go by their train. Domestic type microwave oven you have also having in your home, domestic oven. The in that I am showing you what are the parts or components inside it. One is the microwave generator, which has the magnetron inside it, which is responsible for the power frequency in the microwaves, and it converts also the electric field into the electromagnetic fields. Okay, let's see here, this side all are the, these sides also, these are the magnetrons, and these magnetrons are the responsible for the uh, generating microwaves. Okay, and this is the microwave cavity. Microwave cavity is where we place our samples. Okay, and it should be in such a material so that should be reflecting, it reflects all the microwave energy and throughout no disturbance, because if you it will absorb the microwaves generated, then there is no purpose of giving the microwave into the samples. Okay, and this is where we put our samples. Okay, microwave. Hmm. Manual I am setting it. Okay, okay, time I am giving, this is 100%, power is the 100%. You can fluctuate the power also, means like you can 0 to 100% you can fluctuate. 100% means if the input power is our 70 watt, then 70% of means 100% means 70 watts has to be the output electromagnetic field. If you are giving the 50% of the power, then it should be the 70 of the 50% of the 70, means 30 watt for the magnetic field. Okay, giving time, I'm just giving 1 minute. Okay, start. You can see that that is the processing, and microwave already started extraction. After that you can means take the extraction vessels, and the also the filtration, you can do filter or the centrifuge the all the solvent compounds. Okay, then temperature, temperature is the biggest factor, because in the microwave there is a temperature rise between from the inside to out. In suppose you are heating something on the means stirring plate or hot plate or the how the heating should be done. First the outer vessels, then outer surface to dhire dhire means slowly it will go to the inside. But in microwave the process has to be from inside to outside, because it is employs in the cellular levels, because the it is, I have told the mechanism, due to the dipole and the ionic conduction, the heat generated from the inside or during the cells. So already done. Stop. Then it will cool the machines, then it will be automatically over. Okay, then I will go to the advantages. Very less time, regardless to means regard from according to the ultrasound, it will give in minimum time it will give very higher yield, higher efficiency. The disadvantages is obviously capital cost will be there, investment will be there, but the another means disadvantages is the temperature, because in leisure time the temperature will be very high, so you have to optimize the time so that the temperature rise will be in the minimum, so that your target compound not be in degraded or means reduced. Okay, you can see heating is there, you can see the vapor is coming from there. Okay, the after this you can filter the samples and getting the supernatant. Okay, and this is all about the microwave assisted extraction. Any doubt you can tell me. Okay, now fine, we'll see you soon.